So Ben, I want to look to you for this question here as we dig into some of the R&D and manufacturing innovation needs facing this space. Can you elaborate for us a little bit as to why you think there is such a disconnect, particularly in the RNA-LNP space, between the preclinical models and clinical models, and what types of innovations we're going to really need preclinically to try to overcome some of these barriers?
So if I knew the answer to that question,
we wouldn't be having this panel.
I'd probably triple BioNTech's share price overnight. So
Dang it. I was hoping you'd have the answer for us. How are you chipping away at that question then, right?
So I think, I'll tackle this on some of the screening methods, I think, is the way I'd like to tackle it. So I was at the biggest biotech conference in Australia last week. Funnily enough, it's called BioNTech, and really, the name works.
I love it.
And they had a whole session just devoted to talking about ex vivo models, as a way to do a lot of that preclinical testing and helping make decisions on what you decide to get into phase 1, basically. And so, this is speaking as Professor Ben, scientist, not as BioNTech.
Love it. Own it.
I just because I talk about these things doesn't necessarily mean the company's working on it. But I think organoid models are looking really interesting. So, question 11, there's a few questions about, how can we screen lots of different compositions, and so I'm really curious about the use of different organoid models to be a little bit closer to the real world than just a particular cell line, an immortal cell line, for example. So that's interesting. I'm interested in things like DNA barcoding. So if you can use DNA barcoding in preclinical models, you get a lot more bang for your buck. So you are able to screen multiple formulations in one hit in terms of biodistribution and things like that. Not necessarily potency, but you can sort of get more bang for your buck with models like that. And then there's some other really cool techniques, and this is me just geeking out as a scientist and not speaking as BioNTech. And there's some really cool models using people who've essentially had an organ transplant. So one example is a liver. So you're having a liver transplant, and then there are techniques now where you can perfuse these livers and keep them happy and healthy, inverted commas, for a number of days, sometimes over a week, just out in the lab. So you can actually test a real human liver that's diseased, maybe it's cancerous, with a particular formulation and get a little bit more of a real world indication about what's going on. So things like that are pretty cool. It's not like you can do that every day, right? Like how many random patients' livers will you get access to? but
probably
I
not
think,
a lot. But still.
But that's pretty cool. So I think, I'm not sure I'm answering your question, but it just maybe is.
It's a next gen. We're thinking big. We've got big dreams, right? What can we do, right? What are some of the next step innovations that we can explore? I mean, I'm assuming there are, as you pointed out to one, right? There's some big roadblocks to implementing these on a broad scale, right? But I'd be curious, Kate, I've been watching you take some notes down there. Are you kind of in agreement with Ben here? Are there areas that you're watching or most interested in, right? And seeing further innovations and in terms of how we can improve, right? The screening of these potential products and translate them into the clinic more efficiently and effectively.
Yeah, I think that I love organoid, and especially when you zoom into certain organ, I think even you can use organoid to mimic the patient condition, be able to at least address certain lipid and formulation can get into the cell once you get them to the right organ. But the bigger question, like what Ben said, whether the barcoding, DNA barcoding will help you to actually direct you to the right organ. And I think those are the, people have talked about this for quite a while. Some people use it, and we try it, and it is always some confusing data coming out of that, right? Because again, this is the lipid nanoparticle, and once you formulate, and you have 2 or 3 or 4 formulation coming together, they do tend to mix sometime,
fusion.
and then and make the data hard to interpret. So one of the things I get, if I have a tons of money, and I think using AI, and also unfortunately, I think at the beginning, some of the formulation, after you do very targeted screening in vitro, get into the in vivo study very quickly, and then having that formulation and having that mechanism connected with what you see in vivo, and then be able to come up with some link, help you to improve your lipid structure and also formulation. So I think those are the things people are still playing with. And I have seen some of the AI-driven company come up with something cool, at least faster than what we have seen. So we also try to combine that learning, really limit your question in vitro, and then be able to use massive data to answer that question and help you to get into very targeted in vivo study. Organoid, I think I worked with them before, and it's still very hard to scale up, right?
Yeah.
And so that is that is probably one of the confirmation study you can use.
Okay. I was going to say there might be some regulatory questions as well, right? In terms of this, I think, would there be any regulatory barriers or no?
Yeah, because, deliver something into a particular cell to deliver expected PD function, there's just so many interfaces, right? So make sure the question, the model you use address particular question, the organ, the cell, the endosome release, and the expression efficiency. I think each step probably require different model at the end of the day. If you, I think it's garbage in, garbage out. If you don't really understand any of those, you just, we're not doing science. We're really just study phenomena, right? So, and I think that's why I see the field will go into a large data generation and coming out with some basic understanding of each step, and then circle back. And, I think this is sort of very hard, but it's a sort of necessary pain we have to go through.
Yeah. The accrual of all of the data and then making sense of what that data is telling us, right?
Right. You can only go so much. Yeah, with your blinded. Sorry, Ben.
Go ahead, Ben.
Kate, my question was to Kate, when you talk about the field. So in your mind, how do you define the field? You're talking about the academics out there, the earlier stage research institutes.
I see a biotech and big pharma, actually. I somehow, I see a lot of literature coming from research institute, but I start looking at the challenge to industrialize some of those concepts. I think that's literally what you guys start with, right? So some of the formulation just not scalable. And so I see biotech and pharma working together. I see that as a more active field, because it is come to the scale. It is coming to the practical aspect of this now. I love the new formulation coming out. I love the new chemical coming out, but it just a lot of them is still need to understand step by step, but not just the lipid itself, but really how to get an organ, and then the endosome release
all
and
of the challenges.